Eukaryote, but you're not an animal, or a plant, or a fungus. And, you think a fifth grader knows that? So another way to describe protists is that they are eukaryotes that are derived relative to archaea and bacteria because they have cell membranes and they have nuclei and all those other membrane-bound organelles. But they're not animals, plants, or fungi. APF here. So they have certain ancestral characteristics relative to animals, plants, and fungi. So it's not truly holophyletic. It doesn't include all of and only the descendants of a single common ancestor. So it's not a clade, which means it's not a particularly good kingdom. So we should really divide protists into several kingdoms. But how many? For our purposes, we can say that the protists are something of a problematic kingdom made out of several clades. So what are clades again? Clades, remember, are anywhere we could cut a branch on this tree and have everything fall out. So each of these different colors represents a hypothesized clade of organisms. So in case you're wondering, you are over here under this yellow star with the animals and sponges and fungi and a bunch of other things in a group called the Opisthecons. We share our clade with the amoebozoa. So all of these organisms, different types of amoebas and slime molds. So we could cut the branch here, and that would form one nice, neat clade. The Archaeplastida, we could cut right here, and that clade falls out. Rhizaria, alveolata, stramenopiles. We usually group these three together, and then we've got the excavates and these guys over here. So this kind of looks like a big, colorful mess, because it is. A lot of strange words that you may have never seen before in here. A lot I've never seen before. But we can neaten that up and have it look something like this. This is a little bit more orderly. So this has a lot of the same facets as this, only let's see. So here are the excavates down here. Here's SAR over here, SAR. Here are the Archaeplastida, and then these two bluish clades go together in the Uniconta. Okay? So we've got two monophyletic clades of all protists, and then we've got two paraphyletic clades of protists. Why are they paraphyletic? Because they include most, but not all, the descendants of a single common ancestor. In the case of the Archaeplastida, they're missing the land plants. Land plants are not algae. They're something very different that have evolved out of this lineage. In the case of the Uniconta, also paraphyletic, because it includes mostly, but not entirely, animals. Protists, except for the animals, and except for the fungi. So everything in yellow here is a protist, and these three things in white are called our crown eukaryotes, or our three most familiar eukaryotic kingdoms. All right, so we've got four clades of protists to discuss. What does a clade correspond to taxonomically? Is it a kingdom? Is it a superphylum? Taxonomists still haven't come to a complete agreement on this just yet. But I will talk about these clades just as that, as clades. So just like on the previous slide, we saw that there were these different colors for these four different clades. I'm going to use this convention for our four groups of protists. So the Excavata are going to be this toffee-like color. The SAR clade is going to be this kind of foresty green. Archaeplastida is going to be this olivey green, and then the Uniconta are going to be this sort of royal blue. So 2 monophyletic clades and 2 paraphyletic clades. So the Archaeplasta is paraphyletic because it includes plants. The Uniconta is paraphyletic because it includes fungi and animals. All right, so starting with the Excavata, this is a group of protists that are characterized by having one side that looks like it's been scooped out or excavated. So that's where the name of this clade comes from. And for our purposes, we're going to talk about 4 of the main phyla. Phylum 1, Phylum 2, Phylum 3, Phylum 4, or phyla within the Excavata. So our first clade of Excavata are the Diplomonadida. Try saying that. Diplomonadida. Diplomonadida. Diplo, like we've seen diploid, means 2 of something. Monadida suggests cells or nuclei. And that's how we can recognize members of Diplomonadidas, because they have these 2 equal-sized nuclei. Some folks say they even constitute 2 cells that are fused together. They are all heterotrophic. They have to feed upon other living organisms, either by being free-living, they just swim around throughout the environment, finding things to eat, or they can be parasitic, and they have an intimate association with a host. They have an even number of flagella, either 2, 4, or 8, like you can see in here. They have very reduced or lack in mitochondria. And this lack of mitochondria is something that used to be thought of as an ancestral characteristic, but as we've found out through molecular data, they have had mitochondria in the past, and they have lost them. So that's what I mean by secondarily lacking in mitochondria. They had them and lost them. It's not that they never had them in their evolutionary history. But since they lack mitochondria, they can live anaerobically. They are facultative anaerobes. Many of them have habitat in the guts of insects or vertebrates, especially this particular guy here, Giardia lamblia, now sometimes called Giardia intestinalis, which is an intestinal parasite that lives in a number of vertebrates, especially warm-blooded vertebrates, mammals, and birds. So here's another electron photomicrograph of Giardia. You can kind of make out the 2 cells here, but it's not translucent, so you can't see the nuclei inside. But in this diagram over here, you can see how those 2 nuclei kind of look like eye spots superficially. This is the excavated surface here, which if we go back a couple of slides, they're all attached to the intestinal lining of this organism here, which they're parasitizing. So this structure here kind of is like a suction cup to help them feed. And you can see there are multiple flagella. We've got 1, 2, 3, 4, 8 on this guy. 1, 2, 3, 4, maybe 6 on this guy here. So those are your diplomonads. And Giardia causes a disease called giardiasis, which is often found in a lot of places where sanitation is lacking, shall we say, where water treatment is not practiced regularly. You can get a giardiasis, and I've had it, having lived in the tropics. I can't say it's too pleasant. Causes things like diarrhea and flatulence and nasty sulfur burps. Not a fun thing to have. Our next phylum is the phylum Parabasala. The Parabasala are so called because of the presence of a parabasal body, which is a modified Golgi apparatus. These are also heterotrophic. They don't photosynthesize. And they have a stiffened structure, needle-like structure called an axostyle, which helps give rigidity to the cell which makes up the organism. These organisms have flagella as well. That's how they move. They may have as few as 4. They may have thousands of flagella all over the surface of the cell. Also, very reduced or absent mitochondria, which means they can live anaerobically. Some are associated with intracellular or surface-attached spirochetes. And 2 examples that I'll give you are Trichomonas vaginalis, which causes a sexually transmitted disease called trichomoniasis, and Trichonympha is a symbiont of termites, and it lives within their guts. And you can see both of these start with tricho. Tricho means mesh or thread, and it's because of all of these flagella on the outside of the organism that they get this name. So here you can see a diagram of a member of Parabasala. You can see here's the axostyle here, in this color here, 4 flagella. And many have this undulating membrane. This undulating membrane, which kind of acts like a sail or a keel to help them move around as well. Like you can see here on this colorized scanning electron micrograph of Trichomonas vaginalis. I think this guy down here, who's this guy? I'm not sure, I don't remember. Anyway, moving on to our next phylum within the Excavata. This is the phylum Kinetoplastida. So most kinetoplastids are parasitic. They're all heterotrophic. And our book lumps together the kinetoplastids and the Euglenophyta. So they lump them together into the phylum Euglenozoa, but there is some controversy on this. I'm going to teach it to you like there are 2 different things. The Kinetoplastida are characterized by having a structure called a kinetoplast. The kinetoplast is a single large mitochondrion that has a big mass of DNA. And that kind of makes sense. We can think how kineto means movement, like kinetic energy. Plast means body, like a chloroplast is a green body. So this modified mitochondrion, it makes sense that it would have a name like kinetoplast. One of the most famous kinetoplastids is Trypanosoma brucei, which is a protist that causes African sleeping sickness. African sleeping sickness is carried by this fly called a tsetse fly. The tsetse fly bites an individual and infects them with this trypanosome, which swims between the red blood cells, and it makes individuals who are affected become lethargic, and they become more and more lethargic. They spend more and more of their time sleeping, less and less time in a waking state until they develop paralysis and eventually death if untreated. So kinetoplastids have a flagella that's attached by that undulating membrane, similar to what we saw in Parabasala. But there are some free-living kinetoplastids, like, for example, bodosaltans. So here we can see kinetoplastids in the form of Trypanosoma brucei swimming between, these are all red blood cells in here. And they are able to evade our immune system by constantly changing their surface proteins on the outside of the cell. A huge part of the genome of Trypanosoma brucei is dedicated to this kind of disguising of itself to avoid the immune system. Here you can see that undulating membrane again on Trypanosoma brucei. Our last